Good day, everyone. My name is Charo from Ridge in Taiwan. I would like to talk with you about how to easily use PCR to discover the pathogens earlier and stop the disease outbreaks in your farm. As the latest product of Ridge, POCKIT Central provides a very simple operation. Just put the sample in and the answer will out after 85 minutes. It can help you to save time in doing a strategy, saving your farm in time. First of all, I'll show you the situation of global shrimp production and the outbreaks of shrimp diseases. Then we'll introduce what is PCR, how to easily use it to detect the pathogens. Monitor your farm to control disease and stop the outbreaks. Then introduce our product, POCKIT Central, with the core technology, IIPCR platform, and some shrimp reagents. In the end, I'll talk about the applications for POCKIT Central in multiple fields, even with COVID-19 for human. GAA survey results show that they expect 2020 to be a year of 10% decline with global shrimp production because of the impact of coronavirus pandemic. It's great though to see that 2021 will be a recovery. The total market should grow back 8.4%. For the early years of this bar chart, what's causing this decline from 2011 to 2013? FAO showed that APAN-related declines 2011 to 2013 are expected. For China, minus 26%, Thailand, minus 47%, and Vietnam, minus 43%. These disease outbreaks causing a huge economic losses. Outbreaks of white spot syndrome virus were first found in Panus japonicus in China in 1992. In the last decade, there's two diseases were reported in 2009. Acute hepatopancreatic necrosis disease, APAN, also simply known as EMS, early mortality syndrome. The pathogen was identified as Vibrio parahemolyticus. Diseased shrimp have a pale hepatopancreas, VCMD, which is caused by convert mortality notavirus. In 2010, white feces syndrome has spread through Asia and caused 10 to 15% loss in Thailand's shrimp industry. Some research show that white feces syndrome may be related to Vibrio species and EHP. Furthermore, decapod iridescent virus 1 is outbreaking in China in 2016. How do we do for the farm in the future? Discovering the pathogen earlier and stopping the outbreaks. What you can do is checking the growth signs, performing bioassay, histopathology, and PCR. PCR is a technology with high sensitivity and high specificity. What is PCR? Sometimes a very low concentration of pathogen is hard to be detected. So we use PCR to amplify the nucleic acid of pathogens for detecting it easier. And that's why make this technology a high sensitivity. Moreover, the amplified sequence is designed with a certain region of target DNA, so it can bring on the specificity of detection. The full name of PCR is polymerase chain reaction. It's through 3 temperatures to do denaturation, annealing, and extension. And these 3 temperatures per cycle doubles the DNA molecules. So 1 cycle get 4 copies, 2 cycles will get 8 copies, then 2 billion copies of target region will be obtained from a single DNA molecule after 30 cycles. After that, we could do the detection easily. Traditional PCR usually used to be performed at the laboratory, and it takes time to send sample to a diagnostic lab. Assuming that they have an APAN positive stock from the hatchery. Once onset of symptoms, it usually takes 2.5 days to confirm APAN positive, then start treatment strategies. But for APAN positive shrimp, there would be a high mortality within these 3 to 5 days transportation. Is there any better way to do? And can we run PCR at the farm directly? Our latest product is an all-in-one system, provides an easy way to detect diseases by using PCR and allows to run a on-site test. Instant result within 2.5 hours from sampling. No worry for sample transportation, help you to do the strategy and saving your farm in time. Moreover, you can keep monitoring your farm by detecting the pathogens. The pathogens can be transmitted from tanks to tanks, even farms to farms. To avoid the freight, quality control of each batch of shrimp in the beginning of entry is highly recommended. Every stages in a production cycle are important and need to be well quality control. Checking the live feeds, monitoring on every batches of post-larvae, having health management, and strengthen biosecurity to prevent shrimp from diseases. Meanwhile, the equipment, the personnel, and the transportation need routinely monitoring to minimize the risk of disease outbreaks. To minimize the gap between sampling and test results, on-site PCR is trained for the solution. How to select a proper PCR system for industry. There is a 5A rule. It should be accuracy and affordable. It better be anyone, anytime, and anywhere can do. PCR is a technology with accuracy. Affordable reagent cost is not the only concern, but there are operators' fee, shipping cost, and machine maintenance fee. An easy-to-use system, able to test anytime and anywhere, also be a concern for selecting a proper system. And the fully automatic product of GenRich provides a rapid and cost-effective diagnostic choice. How easy to use with POCKIT Central? First, sampling from risk sources, maybe with swabs or tissues. It depends on the targets. Second, take 200 microliter of sample into cartridge. And the last step, put cartridges into POCKIT Central. After 85 minutes, the results will show on the monitor with positive or negative. Then you are able to do the further treatment. Following, I will like to show you the demonstration of POCKIT Central. As the videos we saw for a total solution of nucleic acid detection, there are 3 steps: sample preparation, nucleic acid extraction, nucleic acid amplification and detection. POCKIT Central combines these two functions, so you just prepare the sample, then put into this automatic machine for the results. And there are only 3 things for the consumables and reagent. Furthermore, it's easy to store at the room temperature or 4 degrees Celsius fridge. About the extraction part of POCKIT Central. It is based on magnetic beads. Silicon dioxide coated magnetic beads will bind to nucleic acid after cell lysis. The next step, we will wash off the impurity, and then the purified nucleic acid will be eluted, and the total extraction time will be 25 minutes. Move to the detection part. We use the technology insulated isothermal PCR, IIPCR. We carry out the 3 temperature cycle by a thermal convection system in the capillary reaction tube, which can save in time comparing to traditional PCR. And following is a short video to introduce how IIPCR works. In conclusion, traditional PCR usually used to be performed at the laboratory and need well-trained personnel to operate it. And it takes time to send sample to a diagnostic lab. For our system, you can easily do it at your farm. And because the single default program, everyone can easily perform it with only basic training. GenReach is dedicated to provide molecular diagnostic products that are affordable, portable, and adaptable for point-of-need application. Our company is located at Taichung City of Taiwan. All the designs, reagents, and machines are made in Taiwan. POCKIT Central has been proven to be useful in multiple fields, including aquaculture, companion animals, livestock and poultry, biosecurity, food safety, and even IVD for human health. All these applications can be used with the same model. The following list is our reagent products line for shrimp. We provide various reagents, including white spot syndrome virus, APAN, EHP, covert mortality notavirus, DIV1, and other reagents. So far, more than 200 reagents to detect biothreat for different industries are developed by GenReach. In aquaculture, mainly shrimp and fish. For pets are dogs and cats. About livestock, the species including poultry, pig, equine, bovine, and luminant. Moreover, we can detect microorganisms, GMO, and adulterations in food. And for human disease, we have the products which can detect C. difficile, notavirus, and we also have COVID-19 reagent. For the last part, the take-home message from this presentation is that, first, PCR detection has high sensitivity and specificity as a good tool to control and manage aquaculture farms. Second, you can easily detect various diseases by using POCKIT Central. And you can easily do the PCR detection and the pond site to save your farm in time. Thank you for your attention.